Yeah.
some students are joining because of a link problem. They could not join early. Okay, let them.
Yeah, yeah, sure.
Yeah. So I think almost I can see lot of
There are a few people
lot of
joining.
students are there.
If you
Yeah,
want to
yeah.
give a few minutes, I mean, you can start
Yeah.
with introduction. Yeah.
Yeah. Yeah. Okay. So, a very good morning to the delegates from India, and a very late evening to Dr. Ramesh Koti and Dr. Charan Ganta. And so I hope all the participants, all the delegates have enjoyed yesterday's wonderful talks. And today also we have two eminent and enthusiastic scientists. So Dr. Ramesh Koti, he is going to deliver a lecture on next generation sequencing, and Dr. Charan Ganta, who will talk about stem cells role in the respiratory diseases. And we are really fortunate to have these young and enthusiastic scientists and today with us. And the first speaker, Dr. Ramesh Koti, into his biodata, so it's really amazing. He has received have received several awards and has done extensive research. It's a tremendous, really, it's fascinating towards this research, I think. And now I request Dr. Kishori to introduce Dr. Ramesh Koti. Over to Kishori.
Thank you, ma'am. A very good morning to you all. It's my pleasure to introduce Dr. Ramesh Koti, Senior Molecular Pathologist.
It's a very low voice.
Volume.
Sorry, just
Ma'am.
come near to the mic. Okay.
It's my pleasure to introduce Dr. Ramesh. Now is it okay, ma'am?
Yeah.
Yeah.
It's my pleasure to introduce Dr. Ramesh Koti, Senior Molecular Pathologist. A molecular pathologist in National Institute of Environmental Health Sciences, North Carolina, USA. So, Sir did his Bachelor of Veterinary Science and AH in University of Agricultural Sciences, Bangalore, India. And he completed his Master of Veterinary Sciences in Veterinary Microbiology, Molecular Biology, and Biotechnology in Chaudhary Charan Singh Haryana Agriculture University, Missouri, India. After that, Sir completed his PhD in Cancer Biology from University of Massachusetts Medical School, Worcester, Massachusetts, USA. And Sir worked as a postdoctoral research associate in the University of Minnesota, Minneapolis. And after that, Sir worked as assistant professor in Massachusetts Cancer Center, University of Minnesota, Minneapolis, Minnesota. After, besides that, Sir received so many honors and awards right from UZ, that is from the Bachelor of Veterinary Sciences. But here I am giving a few of important awards which received by the Sir. He received EAS Gold Medal for first rank in BBSC, and first rank in BBSc and AH degree in 2001. So Sir also received Rajinder Pal Singh Gold Medal, Best Research Worker for MVSc thesis work in 2010. And Sir received C.A. Davis Foundation Veterinary Pathology Resident Scholarship Award in 2012. And Sir also received the NTP Best Paper of the Month in the month of September 2019. So Sir also received the Best Paper of the Year Award from NIEHS in 2019. And Sir awarded with the society. Sir awarded with the STP. Awarded with the Distinguished Early Career Award from Society of Toxicological Pathology. And other important is the award that is a Fellow International Academy of Toxicological Pathology, IITP. After completion of his after completion of his assistant professor, so he joined in research. Sorry, so he joined in the Research Triangle Park, North Carolina, as a staff pathologist. So he worked in that cadre for the 4 years. After that, he promoted as a senior pathologist in the same Research Triangle Park, North Carolina, and he is doing the same thing that is a senior pathologist. Besides he is acting as a on-site contractor for cellular and molecular pathology in this NIEH. So actually, Sir has a very big biodata. So I am presenting a brief biodata. So thank you, sir. So thank you for accepting our invitation for giving your presentation. Thank you, sir. Now I request Ramesh sir present your talk.
Yeah. He got so much gold there.
Yeah.
Very rich pathologist.
Yeah.
Can you hear me now?
Yeah.
The presentation
Yes,
presentation in the
Very clear.
presentation view.
yeah, it
Okay,
is.
thank you.
Yes.
Yes, we can use.
Thank you, Dr. Kishori and Dr. Josna and my colleague Charan for inviting and for putting this wonderful session for 2 days. And so when Dr. Charan contacted me about this, so he basically told me to give a very fundamental concepts in next generation sequencing and its application. So I did not put a lot of data here. So if anybody interested in knowing the data more about those, I'm very happy to send the publication if needed. So as I said, I will be covering fundamentals of the next generation sequencing and what are the lessons we learned for the last over 6 to 7 years by dealing with various platforms of the next generation sequencing. So we all know, being a biologist, we play a significant role in understanding the molecular events happening within the cell to understand the mechanism of disease or mechanism of the entities you're dealing with, whether it is a cancer, whether it is diabetes, any of these entities. As Dr. Kedkar pointed out, even the forensic, like a dietary, and Dr. Sino pointed out about various immunological disorders. So with that, so brief outline of my presentation today, I will talk briefly about history of sequencing. When we go history of anything, any of these techniques, you learn a lot of fundamentals and concepts. So, and I will jump onto the Sanger sequencing, that is the first generation sequencing, and very detailed next generation sequencing, including various steps in the next generation sequencing. And also delve into a lot of issues to be considered during designing these studies and collecting the sample for these studies. So one of the most important aspect of this presentation at various steps, I would list the challenges and the lessons learned. They all come from hands-on experience with this big data. So I will briefly jump into the next generation sequencing applications in biomedical sciences and give some examples from our laboratory.
Excuse me, sir. Sir,
yes.
Padma, you just stopped your presentation, sir. If you don't mind, will you please share your screen?
Let me go back again.
Yeah, screen.
Yeah. Can you see now?
No, sir.
Yes, sir. Yeah.
Yes, sir. Yes.
Okay.
Yeah.
Okay. So, and also as requested by Dr. Ganta, I briefly touch on how we can apply this technology or these assays into coronavirus, very relevant to the current situation. So my objective for the students, so we are viewing this webinar is, at the end of this presentation, if you get, what are the 3 things you would like to know from the next generation sequencing, my objective is achieved. So of course, I will get more into the limitations of the next generation sequencing. All novel technologies they're really appealing. And sometimes they're really a boon to biomedical research, but they all come with their inherent limitations. And there is a lot of scope for misinterpretation, especially when you're using it for the first time. It happened to us, it happened to many. So, and things get delayed because you have to learn from your own mistake. So more than anything, especially for the faculty who are involved in probably serving as a ad hoc reviewer for some of the journals which are dealing with these big data, what are the things you would be looking for when you are reviewing the data from these next generation sequencing? So most of us, since you guys all from Department of Biotechnology, so you know the entire discipline of molecular biology started with a central dogma. So the central dogma, which is a unidirectional residue by residue transfer of information from a DNA nucleotide, double-stranded DNA nucleotide, to a transcribed into a single strand RNA nucleotide, and which is in turn translated into 3 nucleotide based codon to an amino acid. So it was unidirectional throughout the biology, you name any species, with few exceptions. As the rule says, in biology, you will always find exception, for that matter, in any of the sciences. So few exceptions include the reverse transcription, the retroviruses, and the prion viruses. Prions, which also play as a protein mold, which doesn't rely on the DNA and this flow of information. So most of the students and the faculty in this webinar know this famous photo 51 of the DNA, which actually give the fundamental idea to Watson and Crick to get to the double-stranded helix. So the entire hard work of Rosalind Franklin benefited these two guys. So when we say molecular biology, it is only 3 molecules. Until recently, we were dealing, as Dr. Kedkar rightly pointed out, and you should be passionate about DNA. I am kind of disagreeing with him because DNA is a good molecule. Yeah, we should all love. Of course, the life started with it. But RNA is a tremendous amount of information within it. So I'll tell you in subsequent slide why RNA is a good molecule to go after. So this was the central dogma, and nobody questioned and nothing added for a period of almost 50 to 60 years. So nothing changed until recently, when a big class of regulatory RNAs were identified, which includes microRNAs, siRNAs, piRNAs, and link RNAs, and there's so many other short and long non-coding RNAs, which constitute a bulk of the RNA within our cell and which play a pivotal role in regulation of all these 3 events, from DNA replication to transcription and translation and post-translational modification. So when I said RNA molecule, it's really fascinating because of this tremendous amount of regulatory roles, which come very handy as a therapeutic target, and also to understand the underlying mechanism of various diseases. So for all the students, when I dwell much into the molecular biology, it is just this one structure. It is a gene structure. So when I say whole genome structure, which pretty much includes the entire genome, when I say exome, it is the exons of these coding regions of the gene, which are important because they produce the protein. Whereas these introns and all the regulatory elements, which play a significant role in how this protein functions. So entire life revolves around this basic unit of life, that is gene. So with that, history of sequencing started as early as 1869 when the DNA molecule was discovered. At that time, it was not a DNA, then it's a nucleic acid or a genetic material. And of course, in 1953, the milestone in the molecular biology, that's where the discipline actually started, the elucidation of the double helix DNA structure by Francis Crick and Watson. And of course, within just less than 20 years, the first sequencing, the first generation sequencing was proposed and successfully conducted by using autoradiography by Sanger. So it took almost 25 years to complete, and it started in 1990. It took 15 years to complete the completion of the Human Genome Project, with the cost of $1 per base. That is humongous compared to today's sequencing. So in 2020, the sequencing is much cheaper and much faster. So each one of us can have our own whole genome sequence within less than 24 hours by just giving 1 ml of blood and for less than $1,000. So you can have your genome in your pocket and move with it. So that is the tremendous amount of progress happened in DNA sequencing, and that really kind of expedited the understanding of biology at various levels. So this is mainly for students. I'm going much detail. I think most of you are already aware of. So the first generation sequencing is the Sanger sequencing. Besides, there are other methods of sequencing which involve a chemical modification method, and there are pyrosequencing. But this was the one which kind of tested the time, and the people use it extensively, and which leads to given opportunity to elucidate genetic information from varied biological systems. So this uses the DNA polymerase, and this is the sequence by chain termination. So by using a specifically modified nucleotide, dideoxynucleotide, one can terminate the chain randomly in a various length. So based on the capillary electrophoresis and the distance these fragments migrated, one can deduce the sequences in a very efficient manner using the fluorescent tagged dideoxy denucleotides. The entire principle was used in the next generation sequencing, except one chemistry difference. We'll discuss that in detail when I talk about the next generation sequencing. So when I said, why did it take 13 years for the whole genome project, human whole genome project, is because they have to go through this lot of very elaborate procedure of fragmentation of the DNA, and then clone them into either bacterial plasmid, and then individually isolate them and sequence them, and then put them back in a contiguous fragment, and then align them into reference genome. That's why it took 13 years by more than 5,000 people worked on the human genome.
Sir. Sir?
Uh-oh. Okay, let me see.
Yeah, it was like silent for some time, and then...
Okay, can you hear me now?
Yes.
Yeah, sir.
Okay, something happened here. Okay, let's see. Now, can you guys hear me? Okay. So, yeah. So what are the big advantages of this Sanger sequencing? Why did it last for so long? Because it is a very simple method. Very simple method. Anyone can use. It's simply you need a PCR machine, and then you can send it for sequencing, even if you do not have a sequencer. And even till today, it has the lowest error rate. It is considered a gold standard, even after the revolutionary next generation sequencing. If you need to validate any of the single nucleotide variants or any of the mutations in the NGS data, you go back to Sanger sequencing. So, and it has a significant long read length, close to almost 1 KB. Of course, now with the PacBio and nanodrop technologies, one can go even up to 2 KB sequencing, even using the next generation sequencing. But otherwise, most of the next generation sequencing involves short fragments. And one of the biggest disadvantage Sanger sequencing is, if any mutation or any variants which is less than 20% of the frequency, you are not able to detect them. That is one of the biggest drawback in cancer biology to using Sanger sequencing. And of course, in terms of the cost, there's a high cost per base, and there is a low throughput. You can only use one sample in a tube, and of course, that will add on to the cost. So as we will discuss more on the next generation sequencing, this is highly poorly parallelizable, meaning you only sequence few fragments of the DNA and in individual reaction. So that leads to tremendous amount of labor and expenses. So as I said, until last 6 years, our group did mainly mutational analysis of rodent tumors, which are resulted upon exposure to chemicals in National Toxicology Program by using a simple mutation analysis by Sanger sequencing. So they isolated the DNA, used specific primers for genes, especially driver genes like HRAS or beta-catenin, and take out the gel and then purify the PCR product and sequencing and compare that with a reference genome and identify the particular mutation. So even using a simple Sanger sequencing method itself, one can get into a lot of insight into the underlying molecular events within even a complex disease like cancer. For example, in one of the study where in our group we did, which are exposed to a ginkgo biloba extract, So this is in a mouse. So either control group or the 3 low dose, mid dose, and high dose groups, they were exposed for 2 years. At the end of the 2 years, the tumors are examined by the pathologist, and then DNA is isolated. And these 2 genes, just by looking at beta-catenin and HRAS, which cost less than $20 for these mutational analysis, one can see here, especially in the HRAS, you can see the number of mutations are much more higher in control group compared to the treated groups. Whereas in beta-catenin, it's another driver gene, the sequential accumulation or a dose-dependent enrichment of these beta-catenin mutation was striking, and it clearly indicated there is a preferential growth of tumor cells which harbor beta-catenin mutations. So beta-catenin is mutated in close to 40% of the human hepatocellular carcinoma. So by simple Sanger sequencing, one can get a translatability and also what is the main genetic event happening within the tumor. So this slide is mainly for students who are getting into the molecular biology. Please go always learn the concept behind the assay. Don't say DNA comes or isolated from the kits. So I know because of the commercial interest, all the kits have the proprietary solutions, but understand the basic concept behind the isolating nucleic acid, RNA versus DNA, and what are the steps, because each steps will give you an opportunity to improve upon. So I'll tell you a brief story here. When I started at National Toxicology Program 6 years ago, we are using a huge amount of input tissue. So close to 20 micron, 20 sections for isolating the DNA. Today, by paying a lot of attention to each and every step, we are using a 10 micron thick section, one to two section for isolating the DNA from these samples. So you can end up saving a lot of input samples. So I think most of you are using
Ramesh, just sorry to interrupt you. I think your screen got stolen by somebody else. You need to share your screen again.
Okay, let me go back, resume presentation.
I request all the participants not to share any screen because it gives a lot of disturbance. And
the speakers don't have any idea what is going on, so so you guys have to tell me about it.
Yeah.
Can you guys see that now?
Yeah.
Yes.
Yeah.
Okay.
Yes. Yeah, we can see.
Good. Can you guys hear me?
Yes.
Yeah,
okay.
You're good.
So,
Good.
Good. So most of us use the nanodrop, and it was a revolution when it started because you can use 1 microliter and you can get the quantity and the quality of the DNA. You can get the 260 by 280 ratio and 260 by 230 ratio. It is amazing. So what happened in like a few years ago when we used the nanodrop and sent it for whole genome sequencing, so the whole genome sequencing core said, you don't have a mass, you don't have a DNA. It was really surprising for us. Like, we sent 3 microgram of DNA and they're saying no DNA. So one of the biggest drawback of nanodrop is it cannot distinguish a single-stranded versus double-stranded DNA. It cannot distinguish even a free-floating nucleotides due to degradation of the sample. So your quantification of using nanodrop is significantly compromised, especially if you're interested in next-generation sequencing. So my recommendation is use a fluorometric method, a qubit 3 or qubit 2, whichever is available for quantification. And of course, you can continue to use the nanodrop for getting 260 by 280 ratio, which indicates the purity of the DNA, especially if there is a protein contamination or not. And if it is 260 by 230, which gives an idea about any salt contamination or ethanol contamination for your DNA. So more sophisticated way of doing the same thing is using this sophisticated instrument like tape station. Where 10TP, we do have these, which uses a capillary electrophoresis, which again uses 1 microliter DNA, which give a tremendous amount of information about fragment analysis. Similarly, for RNA, we use bioanalyzer, which again gives a lot of information about fragment analysis. So this is one of the examples for the tape station. So if you have, which one of the major advantage of the tape station, it gives a DNA integrity number. It's based on the fragment analysis. So for example, if a DNA is isolated from a frozen tissue with a very intact DNA with a large molecular fragment size, so you can get close to 10 DIN. Whereas if you start going towards the formalin fixed tissue, these integrity number goes down. So this is one of the examples from our NTP bioassay tissue samples. All the blue samples are come from the fresh frozen tissues from the tumor samples, whereas these red come from a short duration fixation, 1 to 2 days fixation, whereas the green one comes from a varying number of days, like 21 days, 71 days, and 94 days fixation in formalin. So you can appreciate drastic change in the DNA integrity numbers, which is corroborated by the electrophoresis electrophoregram year for different samples. So more than that, the quality of the DNA matters, especially for the next generation sequencing, because I will talk about the different steps. If your DIN is less than 2, you will be unable to generate the library. We'll talk about the library in the subsequent slides, but you will fail to generate your first step of the next generation sequencing if you do not have a good quality DNA. So another metric one can use, what we call a DV200, that is a distribution value 200. For RNA especially, you cannot rely on the RIN values because you have to consider the area under the curve, which gives a lot of information for RNA compared to DNA. So if you use a RIN, for example, the RNA integrity number for this sample is 2.3. That means it is not eligible for any of the next generation sequencing. Whereas when you look at the DV200, it's called distribution value 200. That means anything fragment which is more than 200 nucleotide is taken into consideration, which close to almost 73%. That means this sample can easily go to NGS assays. So some of these lessons we learned over a period of time is not all DNA or RNA isolation kits are created equal. Especially pay attention to double-stranded DNA versus single-stranded DNA. And if you're using a formalin fixed tissues, deparaffинization on the slide versus within the tube makes a lot of difference. And as I said, pay attention to each and every step of your nucleic acid isolation, which drastically improve the quality and quantity of the nucleic acid you isolate from your samples. So with that, let's jump on to what are the major difference between Sanger sequencing and next generation sequencing. We already discussed the Sanger sequencing, which uses the DD dideoxynucleotides, which are coated with different fluorophores and sequence 10 to 20 or maximum hundreds of copies to deduce the sequences. Whereas the next generation sequencing, which has multiple steps, so next generation sequencing is sequentially identifying the bases which are added while synthesizing. So that's why it is called a sequencing by synthesis, whereas the Sanger sequencing is sequencing by chain termination. So we'll go to each and every step of these in detail. So the first step of the next generation sequencing is the library preparation. Of course, your input DNA is ready and the quality is assessed. You have the DNA integrity number, which qualifies for the next generation sequencing. So first thing, if you're dealing with the whole genomic DNA, you fragment the DNA either by enzymatic method or by physical method, and then you ligate them with adapters. So we'll talk about briefly in the next step. And once you have this ligation, then you'll go for the cluster amplification within the flow cell. This happens in the sequencer mission. So this is how each of your template is amplified, if not millions, thousands of times in these clusters. So compared to Sanger sequencing, you'll be relying here on thousands of copies of sequencing at a time to get to that sensitivity and to prevent any false positives. And of course, the bulk of the NGS work is rely on the data analysis, alignment and data analysis, and a significant amount of time is spent by the biostatistician to get to the biologically meaningful data. So details about the library preparation. So you have the DNA. So you start with your fragmentation. These are the fragmented DNA. If you use a enzymatic fragmentation, you have these truncated ends. You can repair them and create the overhang to enable for adapter ligation. So your adapter includes the sequencing primers and the indices, which helpful for the multiplexing your samples. And if you want, you can also include what you call unique molecular identifier to prevent any error proofing. So once you have your library, so this is what I meant, the unique sequences or the barcode you can include. So that way you can increase the throughput. You can include as many as in one of the assays, I included close to 200 samples on a one flow cell. So 200 samples in one flow cell because each of those samples have a unique indices. So if you include a unique molecular identifier, you can identify the molecules, especially if you use a PCR for amplification, to collapse those molecules and prevent any PCR-based errors during your variant calls. So during the library preparation, lesson learned is pay attention to the concentration of your input DNA and the concentration of the library, and pay attention to the expected size of the library pool. And biggest problem is adapter duplications. And depending on the chemistry you use, amplification versus captured, you have to rely on unique molecular identifiers to prevent any library preparation step created errors. So one of the example here is, so you can see from the one of the tape station electropherogram here, this is a beautiful, a single 300 base pair library. So this is how your ideal library looks. So, but some of the libraries, you don't see any mass at the expected size. And in some, you get some of these low molecular weight, which are a big problem because which creates a lot of sequencing error. I will talk about that in the next step. So that's why if you have of course, if you have a larger fragments in the next generation sequencing chemistry, those does not create much a problem. So by paying attention to these electropherogram, you can easily prevent any downstream problems with generating these next generation sequencing data. So what is a flow cell? You probably heard about multiple times when the people mention next generation sequencing. So flow cell is a glass slide. It is a simple glass slide, just like a microscope slide, which has its own these cells or channels. Within each of these channels, you have these myriads of oligos. There's a mat of oligos or a lawn of oligos, which are corresponding to your sequencing primer, the P7 and P5 in your libraries. So your template bind to this and then start synthesizing, and the sequencing happens simultaneously. So the next step in the next generation sequencing is a cluster generation, which also happens on the flow cell within this. So when you have a sample, you have an adapter and the sequencing primer. So you generate these clusters. So once the clusters are generating, the sequencing is already acquiring the optic imaging detector, detecting which base is added at each of these levels. And based on the color, you can get what is that base located in that fragment. And based on the indices, you know where is this pool coming from. And based on the unique molecular identifiers, you know whether this is a PCR amplified product or your captured product. So during this step, you have to pay attention. Once the sequence is generated, there are a lot of quality criteria you need to pay attention, you meaning the biostatistician or the biologist together. The first thing is the overall aligned rates. So you'll get a number after alignment, and if the library is good, your alignment is pretty good. Look at this Q30 score here. So all of this red line, which indicates the median quality score, which is in the top of the graph, that means from base 1 to 150, all of them have a very good quality. And this is for the read 1, and this is for the read 2. So this is what I meant by percent Q30. This is the most common and a valuable quality criteria one can use in the next generation sequencing. When I say Q30, which means there is a chance of 1 in 1,000 base calls, there is a error. So as I said, the error rate in the next generation sequence is significantly higher. So if you do not pay attention here, you end up with a lot of false positives. So another criteria people use is called percent base. In any biological sample, ultimately, once you get all of the base calls, the percentage of the base, like 4 bases in the DNA, will be essentially close to 25%. So that means it is a kind of a similar throughout these fragments. So I'll give you examples when a bad sample is there, how this look. And again, you look for the overall aligned reads, like a read 1 and read 2, as in this picture. So how those qualities, and pay attention to those. So one of the example here is, see, this is the library which kind of missed our radar when we are looking at. So there is a 300 base, very small quantity, and there's a small fragment of a 200 base pair, which is not a targeted library pool, which created a big problem in the read 2. Just see this one here. So most of the red are in the orange here are low, and look at the overall aligned read, it is only 17%. And if you look at the read 2 for the percent aligned read, it's only 0.29%. And look at the base call. So this is all over the place. What happened is, because this is not a specific template what you're looking for, so the sequencer is eating constantly the flow cell. So that's why it is reading as a G. G is a black. So it's called a G overrang. So if you don't pay attention, you'll be calling them is, oh, a lot of the nucleotides change to guanine in this sample. So that's why the quality criteria you check after the sequencing, it significantly reduces your error rates. And of course, you heard when you talk about next generation sequencing compared to Sanger sequencing, people talk about coverage or the depth. So what are the accepted depth for various these next generation sequencing? For example, for whole genome sequence, 30x is now it's most commonly accepted depth, and the whole exome sequence is 100x coverage. That means each of the exons are at least sequenced 100 times. And for the whole transcriptome, it varies anywhere from 30 million to 80 million rates, depending on your hypothesis, whether you just want to look differentially expressed gene versus others. So these are the various sequencing platforms. I'm only talking about here in Illumina, so the NovaSeq, which is the most recent and a very, very high throughput sequencer, which can sequence close to 30 human genome in overnight. So you can get an entire human genome sequence in less than 24 hours for less than $1,000. So that's what it's summarized here. You can sequence close to 48 human genome using S4, and there are 500 exomes and then 400 transcriptomes. So as I said, biostatisticians play a big role in analyzing the data from next generation sequencing. So it looks intimidating, but if you pay attention to each one of these are really achievable goals for any biologist and a biostatistician. So your role as a biologist actually pretty much completes at this stage. Once you have a good quality library, good quality sequence, pretty much everything is done by the biostatistician here. And then at the end, variant evaluation processing and interpreting the variants, here the biologist comes back again. So there are several variant callers which are freely available for the biomedical researcher, but when you pick them, at least pick 2 to 3 and then get the concordance in your variant calls between these variant callers. So depending on which platform sequencing you're using, so you end up having these sequences at depth, and then you identify the single nucleotide variants. And depending on whether it is an infectious agent versus neoplasm, you classify them either pathogenic or likely pathogenic or benign or malignant, driver mutation versus passenger mutation. And with all the quality metrics to support your results, you generate a report and give it to the biologist or keep it for the record. And one of the in silico validation of all of this data is by the massive amount of databases like Cosmic Database and ClinVar and other databases which are available. Cosmic Database is the most comprehensive database in Sanger Institute, maintained by Sanger Institute. I will talk briefly in subsequent slide on that. So depending on the, if you are dealing with the gene mutations, you will identify these mutations, and so either point mutation or frame shift mutation, and then of course the chromosomal alterations to decipher your molecular mechanisms. So another confusing two terms you come across is SNPs versus SNVs. So only major difference is SNP is already exist in the population, and these are well characterized, whereas SNV is not well characterized and often linked to a disease, for example, neoplasm or diabetes or other genetic variants, which are not normally existing in the population. So this is one of the most important slides of today's presentation. So if you don't take anything else from the next generation sequencing, understand this. So compared to Sanger sequencing, in the next generation sequencing data, you get a context of the location of the mutation. So what I mean is, these are the possible mutations you can get in your sample, and there is a 5' nucleotide and a 3' nucleotide, which creates the motifs. There are about 16 different types of motifs, and there are 6 types of substitutions. In total, which generates a combination of 96 combinations of these mutation spectra, which results in this unique contribution of a mutation signature, and then ultimately leads to a spectra which would give a characteristic feature for any entity, whether it's a tumor or any of these disease conditions. So let's get to the limitations of the next generation sequencing. As I said, you do introduce bias by template representation by including a PCR amplification. Error rate is still a concern in next generation sequencing that can be curbed by using unique molecular identifiers and then short reads. And of course, you need a tremendous amount of bioinformatics support to deal with this data. So for the study design, so one take-home message from this whole experience is, if you do not have a matched genomic control, whether it is a human population or a rodent population, you will be facing a lot of problem with calling the variants. Always have a matched genomic control. That means if you're thinking about 30 samples, that means you will be dealing with 60 samples. So as I said, data analysis takes the bulk of your time. So have a biostatistician from the beginning in your study design. So as a sample collection, this is a beautiful slide of a liver from a mouse. See, collecting the sample is almost half done for in terms of your collecting the data. So if you take the bulk sample like this, all of this together, you will probably end up with not getting anything, because this contains multiple lesions. One is an hepatocellular carcinoma, hepatoblastoma, and there is a normal liver, and there is an hepatoadenoma. So if you take this as a one sample, you'll end up losing. That's where the pathologists come into picture to identify the morphology, to properly collecting the samples. So with that, applications of the next generation sequencing it's tremendous. I don't think I can list all of it. All the speakers who talked yesterday, they can easily apply this technique to all of their metagenomic analysis or forensic analysis or food forensic analysis, DNA barcoding. So some of these were listed here, whole genomic seq, whole exome seq, single cell genomics. So there are so many ways you can apply the next generation sequencing. So what we used in our lab is a whole genome sequencing, whole exome sequencing, and with a whole transcriptomic sequencing, microRNA, which is an epigenetic component, and the methylation, and the ChIP-seq, which is used for the protein-DNA interactions. So most emerging field among the NGS technology is the liquid biopsy. So you can identify an already existing neoplasm using just a blood sample. So with that, I'll briefly introduce what we do in our group as a molecular pathology. So depending on the sample, so what NTP does is exposes 2 rodent species like mice and rat with varying doses. So depending on the tumors, we need to establish whether this tumor is relevant to human disease or not. So as I said, depending on the samples available, we can apply various assays, anything varying from histopathology to immunohistochemistry. And in between, you can get a lot of these NGS assays to identify the molecular mechanism or molecular carcinogenesis of these tumors. So with the interest of time, I know I'm almost close to 46 after the hours. So let's get to this. If anybody interested in looking at these publications, I'm happy to send them. So one of the striking outcome of this next generational sequencing with a very close collaboration with the Sanger Institute is identifying the mutational signature profiles in the mice tumors, which are relevant to human carcinogen. So this is a very busy slide. I don't want any one of us to understand in a limited amount of time. So which clearly says that there are mutation spectra which are shared between the compounds within the organ and also which are relevant to some of the human tumors. So this is the proportional contribution of those mutational spectra, how the mouse and human mutation spectra are related. So this is the database. If any of our students are not visited this website yet, please make sure visit this site. It's called COSMIC. It is Catalog of Somatic Mutations in Cancer. You can play with this very nicely. I don't know whether I will have time to kind of demonstrate you, but I was planning for that, but I don't think I'll have enough time. But if anybody interested, we can always have a brief Zoom session or something with an interested group to go through this.
Excuse me, sir.
Yes.
Sir, if you have time, please take 5 minutes, sir.
Sure. Okay. I will show that one then. Okay. So this is an RNA-seq. As I said, this is not a data-driven presentation for me. So this is similar setup for an old transcriptomic analysis. So one can depict the data using a principal component analysis. You can see all the blue bubbles are the normal samples, whereas the red are the normal samples, blue samples are the spontaneous tumors, and these are the GBE, ginkgo biloba induced tumors. You can clearly see within the principal component analysis, there is a close clustering within the group, and there is a separation between the group. And of course, you can do the hierarchical cluster analysis of a differentially expressed gene, which would give a directionality to the gene expression, whether it is increase versus decrease. And you can see there are distinct changes between these different tumors. And of course, one can get into the pathway analysis, ingenuity pathway analysis, and then get more relevant information for translatability to human cancer. So, and of course, ultimately, one has to validate all of these changes, whether changes in the DNA, changes in the RNA, at the protein. That is the ultimate workhorse of the, our cell. So if you do not validate at the protein, either using immunohistochemistry or Western blot, most of the data will be questioned ultimately by the biologist. So again, these are some of the publication. One can, if you're interested about the DNA methylation, as I said, with the interest of time, I did not want to discuss all of this data. And this is about the microRNA. You can take a look at those publications. If any of you are interested, I can send you the full copy of these publications to Dr. Josna, and she can distribute to all of you. So as I said, using genome next generation sequencing, you can get a lot of this omics data or a big data, which can ultimately you can integrate into other endpoints to make a biologically meaningful interpretation. So again, you probably got it where I'm going with the application of NGS into the COVID, of course, diagnostic assays. So several NGS panel assays are approved by FDA, including for surveillance and to understand the pathogenesis and to identify the therapeutic targets. So for the surveillance, they identified as close to, they can do close to 48,000 samples per day if they want to, using a NovaSeq platform to surveillance the virus in the entire population for the viral RNA. So for pathogenesis, as I said, one can identify how the different transcripts of the virus is differentially expressed in various organs and how that is linked to pathway alterations and the disease manifestation. And of course, for the pharmaceutical industry, it is the therapeutic targets. One can identify these by using all of these NGS technologies. So with that, I think I already discussed all of these points. Let's jump to the acknowledgement side and also go to the Cosmic database. So this is the Cosmic database. So once you go to the Cosmic database, this is called, can you guys see me the browser now?
Yes, sir.
Okay.
Yes, sir.
So this is this is the Cosmic database. This is the catalog of somatic mutations in cancer. So once you go there, click on a cancer genome, depending on where you want to go. I am a cancer biologist, so I am interested in cancer browser. So I go to cancer browser. So I already opened the cancer browser. So this is the cancer browser, and I am interested in the brain tumor. So central nervous system, brain, glioma, and whichever the glioma you click, and then just click go. So then it generates the list of already existing database for this tumor. So for example, you can click any of these genes and then go to, for example, IDH1, which will give you a particular location of the nucleotide. Here, for example, arginine is mutated to histidine at 132. That is the catalytic site of this IDH1. So this is a hotspot mutation. And
Jyoti, please mute. Jyoti, please mute your audio.
So you can use this, for example, say P53, and open the P53 and look for the where exactly those mutations. And then based on your data, you can look whether it is relevant to human or not. So it's a tremendous resource, a Cosmic database to compare genetic events, any of non-human or human species, but this is mainly the human species. Similarly, there is a protein atlas. You can compare the protein expression using that website. So with that, thank you for all the organizers, and thanks for your attention. If you have any questions, let me know, and I would be happy to take those questions.
Sir, I would like to ask a question, sir. I'm Professor Hema from Department of Virology, Shivaji University. Sir, this is not.
Please go.
Yeah, this is not exactly a question, sir. Actually, it's a submission that I just thought of. Just tell you the experience that I have had with the NGS, sir. I was few years ago, I was trying to work on a virus, RNA virus, which is infecting jasmine, sir, jasmine plant. So then, we could not purify that virus because the amount of virus present in that particular species is very, very low. So how many ever trials we made, we could not succeed in purifying that particular virus. So then I thought I'll go for this NGS because then like this only I've heard about the NGS that it can retrieve the sequences from the entire plant. So then I've given the total nucleic acids isolated by me, and I gave the leaf material also to the Bangalore company, and they charged something around 1 lakh rupees, and they didn't give me any data. They said that they could not get any proper sequences at all. So it was very bad experience. Then thank you so much, sir, for your excellent presentation. It was so wonderful that so I'm really grateful to Jyotsna and her team that it was very, very elaborative and very, very useful presentation. So thank you so much. So now I understand that what could be the problems and all. So somehow that was a bad experience that we could not get data. And as you know, we cannot get the support from bioinformaticians all the time. I mean, that's a problem for the biologists, that we cannot really analyze the data. So I asked them what could be the reasons, but they said so many other things. But at the end result, that we didn't get the sequence at all. I mean, I really wanted to sequence the whole genome of that particular virus. It is 10 kb. So
yeah, see, that's the thing. So your input material, you're simply wasted money and effort, and your, because your input material was not properly assessed for the quality. Whether it's you or the company they were dealing with it, they should have assessed those things. I know we have a misconception that NGS can deal with even a low input like nanogram quantity. Yes, in a purified form, because I used as low as 10 nanogram for a targeted resequencing for an NGS. Yes, but the 10 nanogram should meet the criteria required for the input material. So, for example, as Dr. Kedkar mentioned, if he has to isolate DNA from saffron, say, as one of the students rightly asked, why can't we put it in the upstream rather than downstream? So if you have to isolate that from a tons of saffron or kilograms of saffron, so it will be really, really important to have those quality criteria in place. So I hope a core facility can be created at the university level for the biostatisticians rather than having an individual investigator. I think the core facility can be really, really helpful for most of the investigators to get that support.
yeah, thank you so much, sir. Thanks a lot, sir. Sir, with this facility, after hearing about your presentation, so I'm just wondering that, so there is a kind of virome which is present in the human body itself, that millions of viruses are not known. So we can just get through all those creatures, and then we can get a lot of information about it, sir.
Definitely, definitely.
Thank you so much, sir. Thanks a lot.
Thank you.
Any other questions?
Hello. Yeah, if there are no questions, sir, this is Professor Kalarani, retired professor of biotechnology. Very good morning and my greetings to Dr. Ramesh Koti and Dr. Charan. Sir, thank you so much. It's a very wonderful talk and very informative, and it's a sort of a demonstrative information you have given to the students also. I take this opportunity to profusely thank you and just go through, give a recap of all the points so that the students will recall and then maybe make the best use of it. So the next generation sequencing, they might have heard relatively less compared to Sanger's method. So you really started amazingly giving credit to Sanger's sequencing at the same time, focusing on the limitations associated with that and giving appreciation to NGS. You also pointed out how nanodrop method can stop its elevation, because you have to be careful in the process of isolating DNA. And the emphasis you have made on the quality of DNA while you take up NGS is really very, very important and useful for the students to remember. And it's very good that you started from the central dogma and took them through the recent technique, how best the NGS can be performed with the exemplifications and the associated troubles and the limitations at each phase, how to overcome them, and given at the end the vast applications. Avoiding the manpower requirement and the time and the cost, all those things. It's really conveyed the message in a very appropriate manner, sir. I am sure everybody enjoyed your information and the way you have shared it. I appreciate Hema to enlighten again with some points, and we know she's a close associate to our department, and it's really amazing, sir, to learn more about next generation sequencing. And at the end, you have also touched upon Q30 to tell more about it, and the Cosmic website which the students very much need it, and the way how it should be used and all. Thanks to your time and the patience. We know it is very hard time for you. In spite of all those things and your very busy schedule, you spared a quality time with us. Thank you so much, sir. I appreciate
Thank you.
it. And I
Thank
thank
you.
Josna.
Thank you.
Yeah,
Just
just
a minute.
I just want to quickly add something, or maybe Dr. Ramesh can add to that. So regards to use of nanodrop,
Sure.
In India, we have limited resources. We don't have a fluorescent abilities and all that. So maybe Dr. Ramesh can enlighten us a little bit about how can we still use nanodrop and be able to attain a high quality material that we are interested in. Maybe it's a DNA or RNA. I mean, yeah, see if you can give some input on that.
No,
that
no.
might be helpful.
Nanodrop is good. Actually, you can get a easy way of quality and the quantity. So one thing I would, if there is no fluorometer or the qubit or any other instrument to measure the double-stranded DNA, always ask the company who does the next generation sequencing. They will definitely have one of these. So they can assess the double-stranded DNA. So that way it will give a confidence, because if you don't, as I said, we submitted very precious brain tumor sample from rat for the whole genome sequencing, and they said there is no mass. So it was like really, really painful to hear that. So that's why basically, as Dr. Hema mentioned, rather than going through all till the end, ask them, do you have good quality double-stranded DNA to start with? So it doesn't have to be at your university, but wherever the core facility you send it for the NGS, they will definitely have that metrics to test. So, but don't throw the nanodrop. Nanodrop is a good. I mean, I said it is revolutionized the DNA quantification, because 1 microliter was at that time it was a boon to biomedical science. Keep the nanodrop, but don't rely on nanodrop for, especially for example, I have 1 microgram or 1 milligram, 1 microgram DNA to spare. No, sometimes it is overestimated by the nanodrop. So in our experience, close to 600 samples we tested in nanodrop and qubit, it is in the factor of 10, at least 10. So if you really want to make use of divided by 10, that's your quantity of DNA for your next generation sequencing purposes.
Yeah.
Sir, is there any provision, sir?
What?
Can we have
It is catchy.
Can we have provision to upgrade nanodrop to fluoride? Is there any?
It is reasonable. It is not that expensive instrument, the qubit. So it is reasonably affordable, I believe. But there is a cost associated per sample when you're testing, because it uses the reagent, unlike nanodrop. Nanodrop doesn't use any reagent, whereas qubit uses the reagents. Depending on your sample, whether it is a low input sample versus high input sample. If it is an high input sample, the cost per sample is really low. I think in US it's close to $1 per sample, but in India definitely it will be much lower, I believe, per sample for the qubit. Yeah.
Sir, you said the ginkgo biloba extract you have been used for your studies. So are you going to conclude that this particular extract plays an important role in the inhibition or the pharmacological studies? Are you going to end up?
Yeah, I mean, NTP tests a lot of chemical. I'm sorry, I did not give a brief overview about NTP. NTP tests a lot of herbal supplements also for the carcinogenic capability. So ginkgo biloba is a carcinogenic positive both in rat and mice. So although ginkgo biloba is used as a memory enhancer, immunity booster, and even cancer prevention, but unfortunately in the rodent species, it is highly carcinogenic. So the information was given to FDA, but unfortunately it's still considered as a supplement. FDA doesn't regulate all of those supplements. So it's up to the user's discretion to use or not.
This has to reach the society. Actually, this is the invention of your research work, but nobody knows
Yeah,
that
I mean,
it has been used.
that's the one herbal supplement we extensively worked on various aspects of molecular carcinogenesis, and it is at all level. It is relevant to human, and it is clear evidence for the carcinogenic potential.
Isn't that the one that's supposed to increase the memory?
Yes,
One
yes.
should be very cautious when you take up this type of
Yeah.
I'm so
herbal drugs.
glad somebody he took up the project. And
Yeah. Yeah, Dr. Ramesh.
Ma'am, excuse me, ma'am.
Yeah.
Ma'am, there is a
Yeah.
question in chat box, ma'am. Sir,
Yeah, chat box, sir. Yeah, that's what I was
can we
telling.
search
Okay.
novel long non-coding RNAs through NGS, sir?
So, what can we search novel? Yeah, so that's what in the revised central dogma, it's actually become more famous because of the long non-coding RNA. So, if you use an RNA-seq, you will get most of the long non-coding RNA as part of it, because most of the long non-coding RNAs can be captured using the RNA-seq library preparation. So, when I say whole transcriptome, is not necessarily restricted to mRNA. It is the non-coding RNA, and then your mRNA, and then non-polyadenylated mRNAs also. So when I say small RNA, that will become microRNAs and piRNAs and siRNAs. So link RNAs are included in your RNA-seq data.
Yes, sir.
There is another question from Shraddha Mahendra.
What epigenetic
Can it be done for
can it be done for the cancer typing? So there are a lot of panels which are already approved for diagnosing various cancer, including human breast cancer, colorectal cancer. So as low as 20 genes. So they have a targeted panel which can be used to classify even the cancer, whether it is a basal type or a luminal type breast cancer. So based on the molecular typing, they will be used for different therapy. So for example, brain tumor, and no longer people depend on the histopathology for diagnosis. It's completely molecular typing, whether it is IDH1 positive, whether it has all the TERT promoter positivity. So all of these have a lot of panels. So our ultimate goal is to have a panel of 25 genes for the rodent tumors to screen how relevant to human disease. So to answer to that question, yes, there are NGS panels approved to type the cancer and accordingly treat them with the type of mutations or type of changes they have.
Same person, another question, sir. Can we search novel long non-coding RNAs through NGS?
So that is a good question. Search meaning within the data or in the database. So there are databases for the regulatory RNAs. You can, if you find a novel long non-coding RNA, because as Dr. Hema pointed out, we are not even close to explore the all long non-coding RNAs because they are humongous number within the cell or within different tissues. So you can search if you already known, but if it is a novel, then you have to validate and then identify its function within the biological system in the relevant cell lines or relevant system. Yeah.
Thank you, sir. From Geetanjali, PhD student of our university, as there are inherent errors, what are the precautions we have to take when we use NGS data for restriction analysis?
So one of the easiest and relatively doable is using unique molecular identifiers. So in addition to indices, when I was talking about the library preparation, so if you can use a unique molecular identifier that can be a 6-mer or a 12-mer, meaning it's a 6 nucleotide or a 12 nucleotide, depending on what species you use, so you can restrict your error by comparing the paired end sequence, meaning you will sequence both the sides and also collapse using those UMI, so that way you can prevent the error. So NGS is the biggest limitation is the error prone. So you have to use the UMI, and there is another way. It is called a duplex sequencing. So there you will sequence both the strands of the DNA of each and every molecule in your sample, but that will increase tremendously, almost close to 100 times more than what is the current cost. So it is a cost benefit ratio you have to deal with, whether you can deal with the 1% error versus just 100 times more cost.
Thank you.
Yeah. Thank you, Dr.
Sir, sir, is my voice audible?
Yes.
Yes.
Yeah, I'm myself Geetanjali sir. I'm Professor Kalarani's student. Actually, my question was, when we nowadays all the gene data what we take is from the NGS data, and we take our gene sequences for cloning experiments, right, sir? And before we do cloning, we do restriction analysis using NEB, cutter, and so. So if already it is like it has some inherent errors, and like how many, like how good it is, like sometimes we may get
So
the like
depend.
exact restriction sites, or sometimes we may
Okay.
lose the restriction sites. That was my question, sir, because
Okay.
all
So
sometimes if there are there are some more errors, we may fail the cloning experiments, sir. That was my question actually.
it's an important question, but depending on the species, you should always check the reference strain. Don't go with a people who are deposited in the NCBI database. Always check the reference strain. I believe you guys know the UCSC genome browser. So it is the best site.
Yes,
You can
sir.
go
Yes,
to
sir.
the UCSC browser and use the reference strain. Never depend on individual samples submitted in the database, NCBI database. Rely on your reference strain. If it is a reference strain, the criteria to be a reference strain is it is sequenced at least 30 times. So you have a very less chances of having any error put into the reference strain. So you can confidently use those restriction sites in that reference strain. But if it is a novel sequence and then you have to rely on that, then you have to really check your sequence either by RFLP, the patterns itself, or by sequencing the fragments after you clone. By Sanger sequencing.
Thank you,
See,
sir. Thank you.
Going back to the gold standard.
Yes, that's what I was also thinking. Thank you, sir.
So if there are no questions, it is over to you, Josna. Thank you so much, sir.
Ma'am,
Yeah,
excuse me, madam, there is a question, madam.
Where?
What are the limitations of restriction analysis, sir?
Oh, that is a very broad term. So what do you mean by restriction analysis? So you mean, so again, as Geetanjali pointed out, depending on the sample and the sequence you're dealing with, so restriction analysis using multiple enzyme versus single enzyme, and then again, you have a lot of variables within that. So if you leave a restriction enzyme for a relatively longer time, it will start chewing the non-target sites. So I think that question will really need a lot of input from your side. What you mean by restriction analysis? When you mean by a single enzyme or a multiple enzyme? So yeah.
Yeah. Thank you, Dr. Ramesh Koti, for your wonderful talk. Really, I think we would have planned for offline instead of online, because the time is not enough to clear all the questions. Really, it's a wonderful one, and which is the need of the day. The technique, most of the scientific people are using this, and thank you for giving this wonderful talk. And now I request Dr. Usha. I think we will next move on to the next speaker, Dr. Ganta Charan, and I request Dr. Usha to introduce Dr. Charan Ganta. Usha?
Yeah. It's my pleasure to introduce Dr. Charan Ganta, Adjunct Associate Professor, Kansas State University, Investigative Pathologist, National Institute of Environmental Health Sciences, USA. Dr. Charan Ganta obtained his Bachelor of Veterinary Sciences and Animal Husbandry from Acharya Nagarjuna University, College of Veterinary Science, Hyderabad, and then awarded with a doctoral degree in physiology and neuroimmunology from Kansas State University, Department of Anatomy and Physiology, College of Veterinary Medicine, Kansas, and then moved for the postdoc research associate, cancer biology and regenerative medicine from the Midwest Institute for Comparative Stem Cell Biology, Kansas State University, and is also acted as residency veterinary anatomic pathology, Kansas State University, College of Veterinary Medicine, Kansas, and he has started his career with a clinical veterinarian from Highline Hatcheries Limited, Hyderabad in the year 2002, and then eventually elevated to the present position as an adjunct associate professor. So I'll be telling you the milestones of his journey during the in the growth of professional growth as a professional experience. I request convener to spend some more time because it's a very lengthy biodata with a lot of accomplishments. He has started his career with a veterinary anatomic pathology resident in the year 2011, and then veterinary anatomic pathologist from Kansas State Veterinary Diagnostic Laboratory, a veterinary anatomic pathologist in the year 2012, and then veterinary anatomic pathologist director for histopathology services in the year 2013, and then diagnostic pathology consultant for 3 years from 2015 to 17, and then chief scientific consultant regenerative medicine 2016 to present, and then assistant professor from 2013 to 19 in the Department of Diagnostic Medicine, Pathobiology, Kansas State University, and associate director Midwest Institute of Stem Cell Biotechnology for the year 2019 and 20. Associate Director of Animal and Pathology Core, Center for Excellence for Emerging and Zoonotic Animal Diseases but from 2019 to 20, and as a Director, Comparative and Experimental Pathology from 19 to 20 in the Department of Diagnostic Medicine, Pathobiology, Kansas State University. Associate Professor from 2019 to 20 in the Department of Diagnostic Medicine, Pathobiology, Kansas State University, and Adjunct Associate Professor from 2020 to present, like from the May to present, Department of Diagnostic Medicine, Pathobiology, Kansas State University, and investigative pathologist from 2020 to till today from the National Institute of Environment Health Sciences, Integrated Laboratory System, North Carolina area. So this shows his passion towards the research elevated from the with the veterinarian to the investigative pathologist. I really congratulate Dr. Charan for the accomplishment that you have. And he has also undergone a lot of training programs, and then he has handled many research projects as a PI, principal investigator and co-investigator. To name a few, he has completed his projects in the area of
Usha, we can't hear you.
I think she froze.
I think she was frozen.
Yeah.
I think the CV has become too much long. That's why.
Yeah.
I think the machine is trying to stop it. That's enough.
Yeah. So you have such a big biodata, and both of you have very huge biodata, and it's very difficult to concise about the biodata.
I gave, Dr. Josna, I gave you option, one line.
Yeah, I know.
I keep telling the same.
I know, but no. Let all the students know, because that gives a boost up to the students also.
Indeed.
So that is the main intention of the biodata here. So students will get enlightened. Okay, then. Shall we start then?
Go ahead and
Usha,
share my screen, and then.
Yeah, I think that would be better. I think almost it's the end, right?
Yeah, it's
biodata.
almost.
Yeah, almost. So that's why, we should have some offline program. Usha, connected?
I'm extremely sorry. There's some network problem. So did I don't know where I was there. You have to
So
tell
can I
me.
go ahead and get started?
No, sir. Wait, sir.
There, come on.
Wait.
Yeah, last
So he has a commendable research and
Karen.
yeah. No, I'll
Yeah.
tell for his credit, he has awarded with many prestigious awards and rewards. He has awarded with the Charles Louis Davis DVM Foundation Award for Advancement of Veterinary and Comparative Pathology. And then Dr. Charles E. Cornelius Graduate Student Travel Award from the Society of Phi Zeta Sigma Chapter, Kansas State University. And then first place poster presentation in the Society of Phi Zeta Sigma Chapter, Kansas State University, and Graduate Student Travel Award from the College of Veterinary Medicine, Department of Anatomy and Physiology, Kansas State University, and elected to the membership in the Sigma Chapter of Phi Zeta College of Veterinary Medicine. And then Dr. Albert Brogue Memorial Award, College of Veterinary Medicine, Department of Anatomy and Physiology, Kansas State University. And third prize for the best basis science research presentation at 10th annual graduate research forum, Kansas State University. And he has published many research articles in the reputed journals and acted as the invited speaker in various international forums, and he has played leadership role in department and also in various external committees. So Dr. Charan Ganta biodata tells us very clearly that he has commendable research and teaching experience, and you are the inspiration to all the scientific fraternity, Dr. Charan, and motivation to young faculty and budding scientists. And we are very fortunate to have you with us on this occasion. Thank you, Charan.
Thank you, Dr. Usha. Really appreciate very detailed introduction, and it was really nice, and I'm so fortunate to be able to present today and be able to motivate, if at all possible, a lot of people today. So again, I want to first thank Dr. Ramesh for really accepting this time, and he has done a tremendous job presenting about next-gen sequencing. So with that nice start, I hope I can really continue that rhythm here. So the presentation today is about, it's not really about the modern molecular approaches, it's about how the modern molecular approaches has allowed us to get where we are today. But the topic is more in the area of regenerative medicine. So the title will be the antimicrobial and immunomodulatory properties of stem cells and stem cell derived exosomes in acute respiratory diseases. Again, Dr. Joshna Garu, she asked me to present something with on COVID-19 and kind of respiratory diseases. So I kind of thought I would tailor my presentation something in that direction today. So before going further, I would like to acknowledge some people here because in the end, I might just forget it. So I'd like to thank Kansas State University for providing all the support, and Enzo Discoveries for providing, because I've been a consultant and they have provided some funds to do some of my research. And I'd like to thank Dr. Dori Borges, and she is basically my stem cell mentor. I mean, right now she's currently the Dean of College of Veterinary Medicine at Washington State University. And also like to also acknowledge my colleagues, Dr. Luther and Dr. Orala. And here you can see Naveen. Naveen, he's a veterinarian. He recently came to do his master's in my lab. Yeah, he's one of those very rare finds. He got what he call Lord-made hands. He's extremely a valuable player in the lab, and a lot of the data, some of the data I'm presenting today was done by him. And I also have a PhD student, Robbie Smith, and who is working on a completely different area right now, in the area of platelet-rich plasma and its products. And she is an equine surgery resident. Her name is Dr. Haley Eveler, and she has also contributed some work in the area of antimicrobial properties of platelet-rich plasma. So today I'm going to talk more in the area of respiratory diseases. So to begin with, stem cells, the word stem cells means it's a cell that can replicate itself, and it can also differentiate into many cell types. This is a very simple, basic definition of a stem cell. Again, now the basic definition has evolved in many ways, as you will see eventually in my presentation. If you go back to the history, just like Dr. Ramesh has presented, we should know where we started, right, in the past. It's not like sequencing. This is a fairly new concept. Pretty much like the last 50 years, this regenerative medicine has evolved so quickly, and which is becoming the future of the medicine right now. So the first in 1970s, when they first identified what they call as mesenchymal stem cells, are also called as mesenchymal stromal cells. And then in a few years from there, they identified that these mesenchymal stem cells have immunosuppressive properties, and then they proved it in vivo that in fact they have the in vivo immunosuppressive properties. And after that, again, once they have proven the immunosuppressive properties of these stem cells, the things went pretty fast there. And then they did their first clinical trial with graft versus host disease with a phase 1. And there are several of those GVHD, and there is so much of work has been done after that, so which I'm not going to go into the details, but right now there's a lot of progress made. And if you go to clinicaltrials.gov, you can find hundreds and hundreds of these clinical trials in the area of regenerative medicine. So there are two basic types of stem cells, just to give a bigger picture here. What we call as embryonic stem cells. When we first hear about the stem cells, everybody is worried about this word embryonic stem cells because they come from the embryo, from the inner cell mass in an embryo. So which means there is an ethical concern of using them. So that's why the embryonic stem cells have never become so popular. But then the adult stem cells came where we don't have any ethical dilemma, and the purpose of the repair is really served well because it is shown that these mesenchymal stem cells has immunosuppressive properties and also reparative or regenerative abilities. And also it is shown that these adult stem cells have less evidence of forming tumors when they're injected into the body. And here are some of the sources, like in bone marrow, adipose tissue, umbilical cord, placenta. So are some of the most popular locations where these adult stem cells can be safely extracted from our body. It is a natural phenomena that the regeneration happens. So say for example, if there is an injury in our body, in anywhere, like say here there's an injury, so what happens is say the stem cells can get migrated, say from the bone marrow and they will, through the process of homing, which is driven by chemotaxis, they come to the sites of injury. So what they do is they do two major functions. They do cell empowerment by modulating the local inflammatory area, and also they do the cell replacement. If there's a dead cell, they will kind of take its place. And so this is something that happens in our body, you know, in a normal fashion on a regular basis. So, however, as we grow with aging, this process of regeneration kind of decreases. So the whole area of regeneration has evolved is to kind of give that same kind of regenerate abilities as we age. So how can we intervene to make ourselves heal to the normal state? So there are several mechanisms that are proposed how the stem cells actually work. One is through differentiation into the type of tissue. Although the proof is very less, there is not many studies that to show that it really happened, but that was one of the hypotheses. And another way is through release of a lot of cytokines and other growth factors, which is called by paracrine effects, that's what they call it. And by release of exosomes. So we're going to talk more about this today. That is going to be the focus of the presentation. These exosomes contain what you call these microRNAs. They play an important role in immune response, cell differentiation, cell-to-cell interactions, and a lot more. And there is also another method that was recently published is through the nanotunneling. So basically, as you can see here, this is one cell, and this is like say stem cell, and these stem cells extend these projections into the cell that is dying cell, the cell that needs more energy. And these cells will transfer the mitochondria from the stem cells to the cell that is dying. So through nanotunnels and helps in rejuvenation of these cells. So that is one of the very fairly recent concepts that was identified as one of the mechanisms how stem cells actually does their job. So again, these nanotubes, once they're formed, they not only transfer the mitochondria, they also can transfer other cellular organs like even exosomes and other growth factors as well. So we know the mechanisms. Now, what are the functional properties of these stem cells? It is well believed that they have the immunomodulatory and anti-inflammatory activity. They're involved in tissue repair process through increasing proliferation, increasing migration, and decreasing apoptosis. And they also have the antimicrobial properties. And the main focus today will be on understanding the antimicrobial properties and the immunomodulatory properties. So when it comes to the immunomodulation, I know there are several ways how the immunomodulation actually happens, and this is something which kind of gives you like a short summary how this really happens. So what mesenchymal stem cells do is, say for example here, they prevent the macrophage maturation to become more of a dendritic cell. So this prevents a cascade of cytokine storm-like events eventually down the road. And this is what, when you say immunomodulation, it includes immunosuppression and immunopotentiation, both together. So that's where they use the word immunomodulation. So in case of here, they decrease apoptosis and decrease the respiratory burst in neutrophils. And they potentiate they promote the increase in the number of Tregs, one of the most important immune cell that helps in immunomodulation. And they prevent B cell activation as well, so that there is not too many antibodies that are secreted, and that leads to further downstream effects. So, there are several of these proposed. Besides this, there are other cytokines and growth factors that have other paracrine effects. Coming to the antimicrobial properties, it is shown that the stem cells secrete like antimicrobial peptides, which are AMP, antimicrobial peptides like LL37, that can kill these bacteria like Staph aureus and E. coli. It can also release beta defensins, hepcidins, all these antimicrobial peptides are supposed to be released, are supposed to be loaded in high concentrations in these mesenchymal stem cells. And these mesenchymal stem cells also take signals from this bacteria and also increase the production of these particular antimicrobial peptides, and thus promoting the bacterial inhibition. So with all these immunomodulatory properties, we have lots of clinical trials going on for a gamut of diseases. If you just look at all these diseases, most of these are inflammatory diseases. I mean, besides cancer, if you list any other disease, mostly either autoimmune or inflammatory. Again, autoimmune comes under inflammatory disease. So these inflammatory diseases where the inflammation is out of control can be controlled or modulated by the stem cells. So that's how this functionality comes. It's not the stem cells become another cell. It is basically stem cells modulate the immune system that helps change the local environment and promote regeneration. So now we're going to dive into our topic of this talk today, how stem cells can be helpful in acute lung injury. So here is an example of a cartoon depicting acute lung injury. So if you look at here, this is a microstructure of a unit of the lung. The lung is like, it has so many of these alveolar structures. I should have put a nice picture of how the lung cartoon looks like, but anyway, if you look at the very smallest unit of the lung, it has these multiple of these alveolar structures. These alveolar structures are lined by what you called as type 1 and type 2 endothelial cells, and also it has lined by a surfactant layer that helps maintain the surface tension there, and there'll be few macrophages present in there as well. So this is like a very normal lung. And when there is a injury, when there is an acute injury, the first thing that happens is these. When there is an injury, that means the injury has to be to a specific location. So usually, either the endothelial cells lining these alveoli undergo damage. They undergo either necrosis or damage. Say, for example, with COVID-19 or any coronaviruses, what they do is these type 2 here, these big cells here, they have the receptors for this coronavirus. So the coronavirus will directly damage or kill this endothelial cell that is type 2 pneumocyte, the one that is very important in a regeneration of this thing. So bottom line, due to this necrosis or damage to the endothelium, that triggers inflammation. So where these macrophages gets activated and release all these factors that further cause leakage of fluids into these alveoli. So when these fluids leak, that's where you say you're drowning in your own water, in your own fluids. That means that's when you cannot breathe because of all these fluids are filling, pretty much you're drowning in your own lungs. And also there'll be decreased gaseous exchange because of these fluids, and also presence of this lot of protein deposition. So all this stuff really happens in a very quick fashion and eventually leads to acute phase reaction, which is also called as acute respiratory distress. And now the question, how does stem cells really come and play a role, and how can really how do they really modulate this much injury that caused to the lung? So as I mentioned in the mechanisms, one of the mechanisms is through mitochondrial transfer. So when they do the mitochondrial transfer, it is actually shown that when the stem cells or physically given, they go and they bind with these endothelial cells that are lining the alveolar, and then they transfer the mitochondria into that particular cell that is losing its energy because of the toxin exposure or due to the damage. And when they get the mitochondria back, so they will get re-energized and get back to function. So that is one of the mechanism. And besides that, these mesenchymal stromal cells or mesenchymal stem cells increase what we call as angiopoietin 1, and this, and also fibroglass growth factor 7. So all these factors, what they help is in increase the fluid clearance. So when you say all this edema that is pouring in will be cleared quickly out of the lungs, so that means it is allowing you to empty the fluids out so that you can breathe again, you can exchange, the gaseous exchange can take place back again. So that is another function. And we already discussed about the antimicrobial effects, the antimicrobial properties, because they release these antimicrobial peptides like LL37, beta defensins, and a lot more. And also they will promote these, they will tell the macrophages to undergo phagocytosis as well. So that's how the antimicrobial properties will be exerted. And the anti-inflammatory, which I have already mentioned before, the immunomodulatory properties. So when I say immunomodulatory properties, that means it will decrease all the pro-inflammatory cytokine response and promote the secretion of anti-inflammatory cytokines like interleukin-10, so which will alter the inflammatory milieu there and decrease this, what do you call this, cytokine storm. One of the, I think you may have heard in a lot of the scientific literature about cytokine storm. When there is a COVID-19, there is a cytokine storm. So that cytokine storm can be decreased, can be modulated by the stem cells. And what else? They also have an anti-apoptotic effect, which is another very important factor that prevents cell death. So these are some, I mean, there are still more unidentified, but these are some of those that are very identified and have some evidence to say these are all the functions that the stem cells can exert and cause, regeneration in these lungs. And this is a very old concept that was published back in 2014 in the journal Lancet. Again, it's the same theory, but the one I showed you before was a more polished, but this is like more of a old version of the same concept. But I'm just showing you, how far back we were on trying to understand this process. So now the studies in our laboratory, basically are focused in understanding the antimicrobial properties of these mesenchymal stem cells. So for this one, so there were a few published papers, and this is one of the paper that was published. For us is kind of helps us to understand and to build a background for our story. So mesenchymal stem cells enhance survival and bacterial clearance in the mouse E. coli pneumonia. So what they did was they injected the E. coli and waited for 4 hours, and they injected stem cells into the trachea, and they have seen after injecting stem cells there is decreased pneumonia, so there is more bacterial clearance in the lungs. So as you can see here, I'm not going to go too much detail here into the data, but just to give you a perspective. This is the one where they did not give stem cells here. Here they're given stem cells. When they give stem cells, what happens is the lungs show, this is actually a microscopic section of the lung. So here in this microscopic section of the lung, where you can see there are more cells, here there are less number of cells. Less number of cells means less information. So this is just to give you a 1,000-foot view of what is really happening. So saying that the inflammation is decreased in the lungs after we have given stem cells. In the same way, there is decreased bronchi, alveoli, lavas cell counts. That means there is decreased cell counts in the edema, and there is also decreased protein in these fluids as well. So together saying that yes, there is some effect, some regenerative effect of these stem cells when given into this animal that has an acute pneumonia due to E. coli. And then another paper came out. This is also very interesting. This is published in the journal PNAS. Basically, what they have shown is these human mesenchymal stem cells reduce influenza A, H5N1 associated acute lung injury in both in vitro and in vivo. Okay, so here as well is similarly what they have shown is when they're given stem cells in a matter of days, like in up to 2 weeks or more, in 18 days, you can see there's a decrease in the inflammation, decrease in the number of cells in the lung that was treated with stem cells, versus fibroblasts are like a cell control. They're like not stem cells. When they're not stem cells, there is more inflammation. When they give stem cells, there is decreased inflammation. So this is just trying to build a ground to make you understand that stem cells indeed not only theoretically function, they are shown to have a functional outcome. And then they found out the antimicrobial properties of these mesenchymal stem cells is due to LL37, so which I kind of showed you earlier. Now, back to your mechanism of action. Today, we're going to really focus on understanding the role of exosomes in this process. What are exosomes? These are like the nano-sized. When you say nano-sized, these are, they have a range from 30 nanometers to 150 nanometers in size, almost the size of a virus, I can call that, because I'll show you some examples, but most of the viruses are in the same size. These nano-sized vesicles are secreted from various cell types that contain various biomolecules. As you can see here, some of these biomolecules I'm talking about, like a DNA, mRNA, also called, and there are also siRNAs as well. There are enzymes, there are cytoskeletal proteins, and there are several of these cell membrane markers. So you can see that it has a bilipid membrane layer. So this is like up to 30 to 100 nanometers. They're released from these cells, and they exert a wide range of actions. They increase ATP, they increase survival, they increase proliferation, and they increase migration, they increase the anabolic activity, and they also have immunomodulatory properties. So if you look at how these exosomes are made or within a cell, so basically I'm not going to go too much detail. So basically these exosomes are secreted as little budding vesicles outside, which is called as the microvesicles, or if they release it after fusion with endoplasmic reticulum, then our endosome, and it is called as exosomes. So the focus today is primarily exosomes. These exosomes are ones that travel from the cell that is secreted into the target cell, and the way they enter into the target cell, as you can see here, because these exosomes are bound with a bilayer lipid membrane layer, so they will go and fuse with the cell, and they secrete the contents like these DNA, RNA, and protein. And then these protein and RNA, once they go into the cell, they can enter the nucleus and they can cause genetic manipulation in the target cell. And some of the proteins that are present within the exosomes can also exert a wide range of functions in the target cell. So as you can see, they are like little messengers. Previously, it was thought that these exosomes are like waste products, but then they kind of found out that they have a lot of functions. So, this is just a cartoon depiction. So these are all the cells, and these little dots here that are released are exosomes. So if you take a blood profile, like you have RBCs and you have macrophages or lymphocytes, whatever the cells, and you also have these little tiny little droplet-like structures are called as exosomes, which can be also used, can be profiled in the future. Say, for example, with Dr. Ramesh shared with next-gen sequencing applications, we can profile these exosomes and identify from which cell this exosome came from. So, now there is a lot of value to understand the application. Say, for example, if there is a cancer cell that is coming from the prostate cancer, it releases a specific set of exosomes where it is released into the blood. So if you take a drop of blood and identify this particular exosome using next-gen sequencing, and then you can identify, say, if this came from a prostate cancer cell. I mean, the thing is, we don't have the technology to kind of narrow down which exosome or which particular microRNA that is present inside it is causing it. But I think we are closing the gap. There is a lot of renewed interest in the area as well. So basically, this is like a little graphical or a picture how these exosomes travel from cell to cell. So now here I have an example to show. Think about this world as a human body, okay? And these individual skyscrapers that you're seeing there, these skyscrapers are like cells, like individual cells. So when one cell wants to communicate with another cell, how do you go? Either you take a taxi ride. So when you take a taxi ride, if you look at that, put the taxi ride under the microscope, inside that there are little people inside there that kind of carry the messages from one building to another building, right? So these are all like, I'm just trying to throw the analogy to give you a perspective, what we missed in the past, and how, by going more nano-sized, what we're able to identify right now. So here I'd like to play a little video about how exosomes are made. I thought this is a very informative video. I was gonna share my screen. I hope this is gonna play in this one. I think I should share the screen again. Give me a second. Yeah, just let me reshare the screen real quick.
Yeah, we can still see your PPT file.
Yes, I'm gonna stop that and reshare my screen now. Okay, what I was share is cancel. How about a share a window?
Sir, you have already opened your.
Yeah, I'm gonna share my window, so that's probably easier.
Yeah.
It'll come now. It's gonna go back and it's gonna pull up. You can see that?
Yes.
Yeah.
Okay, good.
Yeah.
So I'm gonna directly jump to the place where we have our exosome production. So let me know if you can hear it or not.
The video is not playing yet.
Is it video, Dr. Charan?
Yes, yes.
No,
it's not playing
moving.
yet.
It's not
Oh,
playing.
sorry. Okay.
Just
You're still in the presentation.
Oh, okay. Now, how do I make this?
Sir, you can stop this PPT and then you can
Share your
again
video.
present now share
Okay.
share button and then try once.
Yeah, that's what I was trying to do. So if you say present, a Chrome tab best for video animation. Okay. Okay, there you go. Okay. It's more technology. Okay,
Yeah,
can you see it now?
now we can see.
Yeah,
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 yes.
Yeah.
Excellent. Perfect. I'm gonna
Play from the beginning.
Yep. No.
Inside
Yeah, I
the
think
cell,
this is
the
good.
cell membrane forms
Okay.
inwards.
Yeah, I'll just start from exactly where we need.
Okay.
Let's find out how exosomes are made. Inside the cell membrane forms inward, forming a special type of lipid vesicle called an endosome. Then the proteins, microRNA and messenger RNA,
Can you increase the
the
volume, sir?
endosome's membrane, forming the exosomes. Endosomes then fuse with the surface of the cell, releasing the exosomes outside of the cell. Due to the observation of this process, historically exosomes were believed to be a waste byproduct of cells. But when researched further, and it was discovered that, in fact, these are the mediating mechanism involved in direct cell-to-cell communication. These produce an anti-inflammatory response due to the communication from the many proteins that downregulate inflammatory proteins. Other proteins will then recruit stem cells to the area that will help unregulate these cells to proliferate and promote organized healing through the regulated process. Then once the task is complete, there are additional proteins that tell the cells when the healing has been completed. These exosomes, with the presence of over 1,000 plus growth factors, are being regarded as one of the purest forms of stem cell therapy.
Okay. So I'm gonna go back to the video. So that's how the exosomes are made. I'm gonna go back to the presentation again. So, okay, that's where we left. All right. So now you learned about how the exosomes are made. So let's see how the exosome produced in the laboratory. So that is something that we do so routinely. Majority of our source of exosomes comes from the adipose tissue. We take this fat and then we grind them up and we isolate what you call as a stromal vascular fraction, basically a cell fraction of the fat, and then we grow them in a tissue culture flask for several passages. While the cells are growing, we collect what we call as a media, which we call as conditioned media, and this media will be collected and saved from multiple passages. And once we have like enough of say we go up to like 6 or 7 passages and we have lots of volume of these conditioned media, we subject that to a series of ultracentrifugation steps. And this is like a very long process. It almost takes like up to 6 to 7 hours during the process. And once we go through the ultracentrifugation, then we finally get what we call as exosomes as a little pellet. Usually it is very difficult to see, but we can kind of see sometimes based on how much we have. And then we go through quality control. We use transmission electron microscopy to see the size. As you can see here, this is a bar representing 200 nanometers, and these exosomes are roughly in the size of 150 to 100 nanometers in size. And then we also do nanoparticle tracking analysis. I think I have a video somewhere to show how it happens. Basically, they just show some light on it and just see their movement. And it allows us to see the size, and we can also be able to quantify the process. Although I'm not an expert in nanotracking analysis, but what I really know is basically it allows us to quantify the exosomes, and we can know what size they are present. So it's more of a quality control and see the integrity of these membranes. Again, we know all these how stem cells has immunomodulatory and anti-inflammatory activities and antimicrobial properties and a lot more. But with stem cells, there are also some limitations that are associated with it. As you have heard in the video, exosomes are basically can serve the function of stem cells, provided if we get that enough threshold concentration of members. So the problems with this using the stem cells is because we have these FDA or ICMO regulations that say you can't use stem cells in a specific way, and there is still a long ways to do. There's a lot of clinical trials that needs to be done before we go further. Another issue will be immune rejection of. I know people say that stem cells can be rejected, but I have seen if we use allogenic stem cells, there is a possibility that some of these cells can be rejected. And another unknown factor even today is how many stem cells that you need to give in order to attain a therapeutic outcome. This is a $1 million question so far because it kind of varies by the person. It kind of varies by the treatment, the type of disease that you're trying to treat. It varies by the age, and it varies by the type of stem cells you're using. So there are so many unknowns than knowns on the doses wise. And also another major issue is the stem cells. When you want to use them, you need to thaw these stem cells because you can't be readily culture them to the point you need to put them in the freezer. When you thaw these cells go into a stage of lag where they don't grow well. I mean, they're not really active in secreting in the necessary proteins to have the therapeutic effect. So it takes a while to really get started. So this is another major issue that we always go through. And again, there are also a lot of cell culture associated complications with this, because with cell cultures, if we use some kind of particular serum, like fetal bovine serum, then it carry these genogenic proteins into the cells. And at the same time, if we try to avoid the fetal bovine serum and use other types of sources of serum, there could be associated effects like mitogenic effects can be, or carcinogenic effects, or the stem cells can turn into tumorigenic cells as well. And these are all the complications with the allogenic stem cells. When it comes to autologous stem cells, besides what we have, there's a lot of huge biological variation, and there is also depletion of the stem cell population with age as well. And if you take the stem cells from one person, there not be enough stem cells to attain a therapeutic outcome. So basically, the limitations are always there. As much as good they do, I mean, there are also some of the limitations that are kind of associated with these stem cells. So taking this into consideration and also understanding, how easy we can attain the goal of regenerative medicine by an alternative methods, not using stem cells, that's where the idea of exosomes kind of came into the existence. So we hypothesize that these adipose-derived exosomes, so similar antimicrobial properties on as mesenchymal stem cells against E. coli and Staph aureus bacteria. So here are our treatment groups. We used cell controls instead of stem cells. And we also isolated exosomes from the stem cells as exosome control. We have just stem cells alone, and this is the dose we have used based on the previous studies. And we have a positive control, and then we have again exosomes at 2 different concentrations. So one is at 2 micrograms per 100 microliter, and then 20 micrograms at 100 microliter. So what we found out was, when we treated when we put these stem cells in a culture plate, and we put these, we grow the bacteria, and then we treat these plates with different treatments, whether stem cells or exosomes. So what we found out was, when we added stem cells to these plates, you can see there is a complete depletion or complete inhibition of this bacterial growth of E. coli in the Petri dish. This is an in vitro assay. Similarly, for exosomes at 2 microgram concentration, we also see some inhibition as well. And then with when we increase the dose to 20 micrograms, there is further inhibition of these bacterial growth, which is almost equal to that what we have seen with stem cells, showing that we are trying to show similar effects of what the stem cells can do, but with a stem cell derived product called as exosomes. So, and another important thing I just want to mention here about exosomes is, these exosomes can be frozen and thawed without any problem, immediately, instantaneously. So that prevents any kind of cell culture issues. And there are a lot of other advantages, which I'm not going to go into the details today, but we can control the dose here more efficiently with exosomes as compared to the stem cells here. So here is just a representative image showing a regular plate, a positive control. And then when we added 2 micrograms of exosomes, we see a depletion in the number of colony forming units. And then after we added 20 micrograms of exosomes, as you can see, this plate became clean. So there is no bacterial growth at all in this case. Again, I just want to mention quickly here, this is a 6-fold dilution. This is 6-fold dilution, and this one is undiluted. We took the broth directly and put it in there, but still we would not see any growth here. Again, we saw similar results with Staph aureus as well, so which is very encouraging, saying that across the board between both the bacteria, we see similar outcomes. So what we found out is adipose-derived stem cells inhibited the growth of both E. coli and Staph aureus, but the embryonic fibroblast did not do that. Again, I kind of did not focus on that. So these are the exosomes derived from mouse embryo fibroblasts that showed no inhibition, but with stem cells showed inhibition. So that's what we are trying to say here is mouse embryonic fibroblasts did not inhibit this growth. So it is not any exosome, it is not any kind of cell, it has to be a stem cell. That is a real idea here. That's what we understood from this particular in control. And adipose MSC derived exosomes also showed significant antibacterial activity with similarities to stem cells. Is everything okay on the background? I can't see you. Okay, can I start again?
Yeah.
Yes, sir.
Yeah. So now we came to here. So yeah, I've got a few more slides and I'll be done. Here is the fun part. So now, what's next? So yes. So what's next here? So in vivo clinical study. So this is a study in progress right now. We are conducting a clinical study in a rodent acute respiratory distress model, and we're going to test out what are the exosomes and the role of exosomes in antimicrobial activity. Will be tested, and also its immunomodulatory properties also will be tested in the in vivo model. Now, so this is really what I want to give some parallels. So drawing parallels. So as you have seen from the previous publications that mesenchymal stem cells reduce influenza A. So influenza is a virus. So just to remind you, it causes a very severe lung injury, as you can see. I'm not going to go explain what it is, but you can see this is a damaged lung, as you can see here. This is a COVID-19 lung from a patient, and it has very similar microscopic changes as you see in an influenza lung. So it's not only that, even come to the mechanism of action, what SARS-CoV does is once it's ingested or once it gain entry into your respiratory tract, these cells that are lining the nasal epithelial secretory cells and bronchial type 2 cells are the ones that has this receptor called as ACE2, which is an angiotensin II receptor. When these SARS-CoV virus goes inside the body, what happens? It's going to go and bind with this spike protein. It binds to the ACE receptor and gets invasinated, gets sucked into the cell, and then it releases this RNA, and then it will make more copies of this virus and releases. What it does is recreation of its species. But what really happens is this is where our body kind of overreacts, is what I can use the word, the right terminology. It overreacts because when this ACE enzymatic function is decreased, it increases the pro-inflammatory angiotensin II function that will trigger a massive pro-inflammatory cytokine response, also called as cytokine storm. The viral damage, the damage that is caused by virus, is if it is like 10%, but the remaining damage to the lungs is caused by the cytokine storm, mainly because of release of all these cytokines and other growth factors that causes a lot of oxidative damage and kills the tissue in that location, cause necrosis of the lung. So again, we talked about all the how these stem cells can cause or repair these damaged lung. So here also there is a hypothesis saying it can have similar effect. In case of non-viral induced acute lung injury, we talked about how stem cells decrease inflammation, cause immunomodulation, how the increased alveolar clearance can be done, how the mitochondrial transfer helps in potentiating. Similarly, it is hypothesized that even with SARS-CoV-2 infection, we can have similar outcomes because we're not really trying to treat the virus. What we're trying to is we're trying to modulate the inflammation that is created by the virus, which is similar to a non-viral induced acute lung injury like a bacteria. Even with influenza, acute influenza, that's kind of similar pathogenesis happens. So ultimately, it is like drawing parallels from what we learned from the previous studies here, saying that yes, pneumonia can be caused by H5N1, influenza A can be treated with stem cells. Yes, we can definitely treat the SARS-CoV-2 induced acute respiratory distress syndrome with stem cells as well. So of course, there are a lot of clinical trials already registered, and there's a lot of studies going on already right now. Now, another just a fun fact I want to throw out there. If you look at the way these two look like, can anybody guess which one is a virus and which one is the exosome here? The left one, what is the left one? Is that the virus or the exosome? And the right one, is that the virus or the exosome?
Hello.
Yeah, sorry, I just opened up. Oh, go ahead.
So the left one
The
is
left
the
one is
exosome and the right one is the what is virus.
Yes, so
no,
there you go. So I'm going to reveal the answer. The left one is the coronavirus, and the right one is the
Right is
exosome.
exosome.
Yes. So what the reason is, I'm just trying to draw parallels in multiple ways. So basically, these exosomes has a very, very similar structure as coronaviruses, as the viruses. In a way, I'm not saying that these coronaviruses really, basically, they almost look like one of our cellular organelles. They're nothing anyway different than anything that we have in our body. Just that they're like bad cellular organelles that enter our body and damage our body. Because exosomes also does a similar function. They go into another cell and manipulate the genetic material. And coronavirus does the same thing. They go into your cell, manipulate the genetic material. I'm not saying they're both the same, but they have some kind of parallel functions that has some similarities. Again, the way they enter the one cell into another cell. So when these coronavirus enter by attaching spike proteins, releasing their viruses and replicate. Same thing with exosomes. They also release they can go into the two different ways. They release their cell contents into the cell, and also they get engulfed as well. So there are kind of like a parallels in not only in the way they look like, and also the way they function as well. So, and in addition to that, this is an electron microscopic picture of coronavirus on the left and exosome. They're almost also of the same size. They both are approximately 100 nanometers. I mean, not all viruses are 100 nanometers, but I'm just saying about coronavirus is almost the same size. But a lot of viruses fall in the range of that in the nanometer size. But they all have like similar architecture in having a membrane protein with the nucleic acid inside. So coming back to drawing parallels, again, um, uh, these viruses are basically, imagine that yellow car that is going is like a virus. It's like a bad virus or a bad organelle that communicates from one cell to another cell and transfer bad information and also replicates into another cell and create some kind of bad and cause a destruction. So it's like a coronavirus, the same thing. Again, here I'm just for fun here. I'm just trying to draw parallels here. So ultimately, what I meant to say here is we have a very bright future with exosomes as future one of the future medicine opportunities. Where they can be used for a wide range of diseases.
Madam,
So,
Kalarani madam, madam, can you mute? Yeah.
okay, so with this, I'd like to thank you all for attention and like to take any questions. Hey, Ramesh.
Hello. Yeah. There are a couple of questions.
Sure.
Dr. Ramesh, you need to unmute your mic.
Okay. Okay. Thank you. So, any long-term usage of stem cell often results in cancer. What about exosomes? Is there any possibility of exosomes leading to a cancer outcome?
Yes, there is a possibility in every way because right now we don't know. So it depends upon what kind of exosomes, what is the source of exosomes. Really, we just need to do more clinical trials to understand that process. Just like stem cells, we are still just scratching the surface here. So we don't know. That's a good question. Again, giving that, I want to add one more thing to that. The reason, if you ask me this question like 1 year past, I would have said, nope, it won't. But now I cannot say that because in the past we thought go into the body and they will proliferate and they become tumors. But now we learn how stem cells actually work. It's not by proliferating in the body because stem cells, once given, they will disappear in 2 weeks. That's the maximum time anybody could find a stem cell in a body after they're given stem cell therapy. So they don't undergo engraftment. They just release those paracrine factors and exosomes, and they will disintegrate. And the outcomes are mainly driven by the factors that are released by the stem cells. So now that you ask me that, okay, is it? Yeah. And now I have other concerns right now because learning more about exosomes, yeah, maybe not. Exosomes has more functions, are similar to, I mean, parallel functions as stem cells. So yeah, right now we don't know. So I have some
Any questions from the
questions.
students? Yeah.
Yeah, sir, good
Yeah.
morning. Myself Geetanjali again. Yeah, sir, my
Hello.
question is like, yeah, sir, is my voice audible?
Yes, very
Yeah.
very audible. Yes, I can hear you very well.
Yeah, sir, my question is like, how do we target these exosomes, like by like a cell surface receptor based or any other mechanism? Because if we have to target cell specific or organ specific. Because
Oh,
if
again,
they are non-specific, again they can go and bind anywhere, and they may have negative effects also. What we expect, right? So how
yeah.
we are
I
going to
mean,
control the
uh,
transfer?
yeah. See, again, we are back to again the same thing. We still don't know that question to answer right now. The way to control that is, say, for example, stem cells derived exosomes, which means you're growing a population of cells in a petri dish, and you're collecting exosomes from that cell only. So now you know all the exosomes are coming from the stem cells that you have it. But if you try to poke a drop of blood and collect exosomes and try to identify where these exosomes came from, if you can figure out where each exosome came from, I think then you'll be provided with a Nobel Prize for sure.
Sir, I
That's the thing.
think
I mean,
if
that's
it,
where the diagnostics component comes into play. That's
yes,
exactly the thing that people are trying to unravel. I mean, I don't know that field, but if you go to, there is a society of exosome biology where they have already identified specific markers of exosomes for prostate cancer right now. It's already in clinical trials, and there's a lot of studies going on in the area, also for other cancers as well. So basically, they can take a drop of blood and they can identify specific microRNAs within the exosomes, again by use X-gen sequencing and all these kind of things to kind of identify the specific biomarkers, and then they tell, okay, this particular microRNA came from this particular cancer cell. So that kind of work is already in progress.
yeah, sir. My addition to that is like, sir, if we purely depend on culture-based production of exosomes and identification and trying to understand their activity, if we start synthesizing like in the laboratories, aptamer-based proteins, mRNA, if we can synthesize, pack them in a liposome with a specific marker, we can rather like we can synthesize in large amount, and that scaling up also is very easy, no, sir?
Yes, yes, of course. That's really,
And
it's all about the
safety
next
also
thing.
we can
Control the safety as
safe.
well. Yes.
Yeah, yes, sir.
Yeah, I mean, at this point, we don't have that level of sophistication. If you have to say that, because when you say exosomes, they just don't pack one type of molecules. They pack lots of molecules in there. Even today, we don't know what those are. What are all the components in there? So right now we're kind of relying on just the nature, what nature gives us, and just taking it from there and using it. If you can even understand even further what's inside these stem cell derived exosomes, then you can control and you can synthesize artificially and you can take it to the next level. Again, like I said, it's like deep, deep science. We are not there yet, but I'm sure one day you will be able to chase that.
Yeah, thank you, sir. Very exciting talk. And exosome, first time we are hearing the word actually.
Nice. I'm glad.
Really, really, sir. Yeah, first time. Thank you, sir.
Yeah.
Hello.
So any other questions? So I see here there is in
Yeah,
the
somebody is
chart.
on the line.
Can you hear me? Sir, can you hear me?
Yes, yes, yes. Go ahead. Yes.
Sir, actually you are talking about the different cytokines released during infection of SARS-CoV-2.
Mm-hmm.
So have you identified any specific cytokines which caused damage in the lung? Or these cytokines can be used as a marker for future diagnosis or something like that?
No, not really. Yeah, sure. But thing is, there are no. Okay, according to some literature or what's found out is there is nothing. There's not a specific cytokine that
Okay.
is kind of targeted towards just coronaviruses or any other thing. Whenever there is a tissue injury, think about this virus-induced attack as some kind of injury. It doesn't have to be like a coronavirus. It can be anything.
Okay.
And then the after effects of the injury is release of these cytokines and causing all this tissue damage is what we are saying.
Sir, can
Now,
we use this? See,
if
you have been telling about that they cause damage. Can these stem cells can be used to block the cytokines during this damage or something like that?
yes, that was the hypothesis. Yes.
Okay, that only the hypothesis.
Well, there are clinical trials going on. Again, I said there are drawing because this is a fairly new virus, and it takes time for us to completely understand. They have shown this thing in other viruses like influenza. They have shown with other kind of acute lung injury models. So now they're doing these studies already in with coronavirus, and there is some preliminary studies showing that there is promise. But again, we only are just in months into the studies. It will take a couple of years from now. We will see how this is outcome is going on. So
Yeah. Thank you, sir. Thank you, sir.
no problem. I can go through the chart window.
Yeah.
I'll see.
Rekha.
So basically, it says he says, please, can you find exosomes containing RNA biomarkers in saliva? Yes, the answer is yes. Every fluids in your body has exosomes in it. So yes, we can. But like I said, we are still far away in identifying what those biomarkers are going to be. So that is something requires more modern molecular approaches to understand what's inside these exosomes. Okay.
Yeah.
Any other questions?
I think chart
One
box.
of the question was on whether we can use exosome as a prognostic marker for cancer. I kind of answered that without your permission.
Yes, sir.
So that is a liquid biopsy.
Liquid biopsy, you're right.
They predominantly focus on the circulating cell-free DNA, but people are exploring other options because circulating cell-free DNA may not give idea about the cell of origin. But there are some recent trends about microRNA and some of the protein which are attached to those microRNA giving ideas about the cell of origin. So there are some preliminary findings on identifying the potential cell of origin of those by using liquid biopsy.
Yeah. And there are hundreds of these microRNAs, so there is a lot of profiling that needs to be done. Again, it needs next-gen sequencing.
Yeah. So if there are no questions, I'll conclude the session.
In case of, there's one question here. It says, in case of lupus, can exosomes be taken into consideration for any future treatment? So the answer is, for any kind of autoimmune disease, yes, exosomes can be used as exosomes in general has immunomodulatory properties. So it can be lupus, or it can be osteoarthritis, it can be ALS, or it can be any other autoimmune disease.
Sharon, you're frozen.
Yeah. Charan?
Some network problem.
Let me call him.
Hello, sorry.
Yes, we can hear you in a different.
I was running on a computer battery for the last 4 hours now, so I got this.
So,
okay.
I'm very it did not stop in the middle, at least.
Yeah.
So if you have any questions, they can email.
A small question.